Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Overcoming the Challenges of Immuno-Oncology Biomarker Discovery, From Sample to Analytics. My name is Vicky, and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes, with time for a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank Personalis, who helped develop the content for this presentation. Personalis Inc. provides advanced genomic sequencing and analytics for immuno-oncology. Their mission is to support cancer immunotherapy development by helping drug developers identify neoantigens, understand immune escape, and elucidate the tumor microenvironment. Their patented accuracy and content-enhanced technology achieves augmented coverage of difficult-to-sequence genomic regions, allowing Personalis to provide genomic data of the highest quality and accuracy to help biopharma companies drive their immuno-oncology clinical and biomarker discovery programs. And now I would like to introduce our speaker for today's event, Dr. Erin Newburn. Dr. Newburn joined Personalis as a field application scientist in 2013 with over 12 years of research experience in the areas of molecular biology, genetics, and biotechnology. Erin leads the field application scientists team, which has the responsibility of providing both pre- and post-technical support for the Personalis ACE cancer portfolio. Erin completed her postdoctoral training at the National Institute of Mental Health, investigating candidate susceptibility genes for major psychiatric illnesses. Erin received her PhD from The Ohio State University in Integrated Biomedical Science as a Presidential Fellow. And now, without further ado, I would like to hand the mic over to our speaker. You may begin when ready.
Thank you so much, Vicki, for the introduction, and thanks to the audience for joining us today for the webinar. So let's go ahead and get started. So if we consider both immuno-oncology translational research and neoantigen vaccine clinical trials, having an understanding of the very complex interplay between the tumor, the microenvironment, as well as the neoantigens present is needed. And also having powerful genomic and transcriptomic tools that are both accurate and comprehensive is also needed. So in my presentation today, I'm going to be discussing our platform, ACE ImmunoID, and how at Personalis we really envision this platform as a ubiquitous solution for the development of both personalized vaccines and also to fuel immunotherapy biomarker discovery. But today I'll primarily focus on the work we're doing with our customers who are searching for these more accurate biomarkers for immunotherapy response. So as we've seen, PD-L1 expression is not a perfect metric for determining patient response for checkpoint inhibitor therapy. While it does provide some clinical utility, most are going beyond PD-L1. And also, likewise, mutational burden is an additional metric suggested for determining patient response. It does not entirely delineate the responders from the non-responders. And so there are additional diverse biomarkers out there being suggested. And here are a few recent publications that have demonstrated additional metrics such as gene expression signatures and copy number variant analysis that may prove useful in understanding patient response as well as resistance to immunotherapy. Now, before we continue, we have a quick poll question for the audience.
Thank you for that, Dr. Newburn. And the poll question should be launching on your screen right now. And that poll question is asking, which of these is the greatest challenge for your biomarker discovery program? And the options we have here are difficult or limited patient samples, unreliable assay coverage and missed targets, dealing with complex data, or lastly, timeline pressures and long vendor TAT. And if the audience could please get their answers in, this first poll question will be closing shortly. And once again, this poll question is asking, which of these is the greatest challenge for your biomarker discovery program? And it does look like majority of the audience has voted, so I'll go ahead and close this poll question and share the results with everyone. And it looks like majority of the audience with 38% voted for difficult or limited patient samples. After that, it was a tie of 27% between unreliable assay coverage and missed targets and dealing with complex data. And then lastly, 8% with timeline pressures and long vendor TAT. And with that, Dr. Newburn, I'll hand the presentation back to you.
Great, thanks, Vicki. So as the search continues for more accurate biomarkers for patient response to treatment, I'd like to walk through a few of the key challenges that researchers are facing in this process and also give you some examples of solutions to address these various obstacles. So let's discuss one of the initial challenges, which I believe the audience also mentioned as well in the poll question, which is the ability to work with a more difficult sample starting material like FFPE in your DNA and RNA sequencing process. So, of course, these are very critical, precious patient samples in biomarker discovery. And traditional processes often require multiple samples to be sent to multiple vendors to get the data needed in your study. So, for example, perhaps sending a blood sample for HLA testing, a tissue section for expression analysis, and then sending another tissue section to look at mutational burden. So utilizing a partner that has a simplified process for simultaneous extraction of both DNA and RNA from the specimen, and also using assays designed to work in a sample-sparing manner is essential. So we've designed our approach, our ACE Immuno-ID, as really a one-stop solution that enables multiple analytics from a single sample preparation. So I'll be discussing these multiple analytics further in the presentation, but you'll see that with our high-content assays, we provide a very rich, a very broad data set for discovery. So FFPE samples are the most common sample type, sample format available from tumor biopsies. And while fixation, of course, can greatly preserve your tissue, even preserve it for decades, but the fixation and the chemical modification can degrade both DNA and RNA. So there are several fixation behaviors that can negatively affect the nucleic acid quality, and I have some of those listed here. These can include the time, the duration of fixing. So fixing samples for over 24 hours has been found to be detrimental. And fixing should also occur at room temperatures, as higher temperatures are also not recommended. And laboratories can help and provide specimen preparation guidance for working with FFPE. So for example, at Personalis, we have a guideline document for best practices, and you can see this here on the slide. And what we recommend is using a freshly prepared 10% formalin and PBS solution and fixing overnight. So how do you then assess the sequencing quality coming from FFPE samples? You know, what type of metrics can you use? And so there are several metrics to help you determine the quality of the sequencing data for both DNA and RNA. So to assess DNA sequencing results, metrics that can be considered in this evaluation are, for example, looking at the percentage of mapped reads or how many reads align to the reference genome, looking at the QMAP score, and this shows the probability that a read is misplaced in mapping, or looking at the Q-base score. And this is the probability score of an error in base calling. So as you can see from this recent project of FFPE samples that we performed at Personalis using our enhanced whole exome, the DNA sequencing quality was quite high here with these samples from utilizing metrics such as these in the assessment for quality. And in our lab, to date, we've received a variety of different FFP samples, and shown here is a snapshot of some of these. And as you can see, these materials can come in a variety of different qualities and quantities. And this is, frankly, the reality of working with clinical FFP samples. And in our lab, we've optimized to work with these more challenging sample types and have currently a greater than 95% success rate in those samples that we've received for freshly prepared specimen from our clinical trial partners. And so now that you've optimized and prepared the nucleic acids from the tumor specimen, another challenge is to be able then to comprehensively and accurately identify the potential targets present in those tumor biopsies. And this really begins with improving the assay used in the process. And so for biomarker discovery, broad assays, high content assays such as whole exome and whole transcriptome sequencing are being used. And unfortunately, the issue with conventional assays is that standard whole exome and whole transcriptome assays have gaps and inconsistencies in coverage. And for example, we've demonstrated this here by showing you a sequencing plot of the STK11 gene. And this was using a standard exome assay. So this plot shows you all the exons concatenated together here on the x-axis, and then the read depth is shown here on the y-axis. And if we look across this gene, so gene-wide coverage, you have what I like to call peaks and valleys in sequencing coverage. You have areas very sufficiently covered at high depth, greater than 150x, but you also have suboptimal regions. And as you notice here, even though, again, this gene was sequenced to very high depth, it still fails to cover whole exonic regions using a standard exome assay approach. And variants residing in these regions, this is kind of shown here by the red bracket, could be completely missed using a standard whole exome sequencing assay. And if those regions, happen to harbor a variant that results in an immunogenic epitope, it would be missed by the conventional assay. And so this is what we have really addressed and recognized with our proprietary ACE approach. And ACE stands for Accuracy Content Enhanced. And the way we do this is we use a standard exome as our foundation. So you have broad coverage of all 20,000 plus genes, but we then went back to those problematic areas in the genomic architecture and we supplemented. And we do this in two ways. So number one, by optimizing the probe design, but adding more probes will not completely solve the problem. But number two, what we also do is we optimize the chemistry of the NGS library preparation. So changing the enzymatic conditions, the times, the temperatures to capture those difficult genomic regions. And the standard exome really takes a one-size-fits-all approach to the genome, whereas our capture approach has been optimized to capture those more difficult areas. Plus, we've created and designed the ACE process in a sample-sparing manner, so this doesn't require additional materials. And also the library preps are done in parallel and ran together on Illumina instrumentation, so not requiring additional time either. So we combine these enhancements into a single integrated high accuracy data set in the end. Again, if I go back here to STK11 gene, this example, with a standard approach here, you have these kind of peaks and valleys across the gene and inconsistencies in coverage. And then this is a nice visual of the quality of the data that you would receive from an augmented exome approach such as ACE. So we provide you with the blue, which is the standard coverage, plus the green supplemental coverage here in this plot. So our goal is to really achieve more accurate, more uniform sequencing coverage gene-wide. So the gaps, again, where you had little or no blue coverage in the depth, are now filled in by our ACE enhancement. And so accurate whole transcriptome sequencing is also critical as you're defining your solutions in biomarker discovery. And we apply our same ACE technology to our solution for whole transcriptome sequencing. So as mentioned, in many of these oncological studies, FFPE samples are the materials available for analysis. And in the past, these sample types have been quite challenging for genomic, but especially for transcriptomic studies. And typical enrichment methods for RNA-seq, like using poly-A or ribodepletion, have known limitations, such as inconsistent removal of ribosomal RNA, or also just the inability to work with more degraded materials. So methods such as an exome capture enrichment, like the ACE cancer transcriptome, can allow you to overcome these limitations. It allows you to work with a more degraded sample starting material. So in our approach, we use our optimized ACE probes targeting the exonic region, so multiple ACE probes across the gene with our optimized chemistry to enrich for the messenger RNA. And just as I showed the STK11 example from whole exome data, here with our transcriptome coverage performance, you see the same augmented, more uniform coverage in the bottom of the slide, again with the blue plus the green. So we really strive for uniform coverage across the transcript, which hopefully results then in increased sensitivity to detect variants. So this exome capture enrichment process was specifically designed to improve the performance with FFPE. However, it's applicable for various sample types: fresh frozen, fine needle aspirate, blood, PBMCs, and we routinely accept all of these sample types at Personalis. And so data generated from an exome capture enrichment protocol is very clean, and that's hopefully demonstrated here on this slide, where what you're seeing is that this results in the majority of the bases aligning to the exonic region as compared to a ribodepletion method. And this blue color here on the bar chart shows that the majority of the bases are mapping to the exonic region with ACE. And when you compare with a ribodepletion protocol on the left, you have a large number of off-target reads. So this approach focuses on the regions of interest for more complete, comprehensive detection of the expressed variants. A critical step in biomarker discovery is also gaining an understanding and an assessment of the sensitivity and specificity to identify these variants. And at Personalis, we've done a rigorous analytical validation on the ACE ImmunoID platform, both on our enhanced exome and transcriptome. And in our approach, we've acquired FFPE samples in this validation, as well as we used variants from a dilution series of well-established cancer cell lines to investigate limits of detection for variants. And we were able to demonstrate very high sensitivity and specificity for SNVs as well as indels and gene fusion events, both on our exome as well as our transcriptome, down to a 10% minor allele frequency. And this is an overall summary of those results from our approach. And here in the table, you can see overall sensitivities are shown 98% for SNVs and 95% for indels with our exome, and then also 97% sensitivity with our transcriptome for small variants and 100% for gene fusion events. So this was a very complete and very robust validation, as the results that I'm showing you here represent from looking at over 16,000 SNVs and over 700 indels. So these were very strong results. Now, before we continue on, we have one additional poll question for the audience.
Thank you for that, Dr. Newburn. And that second poll question should be launching on your screen right now. And the poll question is asking, what is most informative for your studies? And the options we have here are neoantigens, tumor microenvironment, or lastly, general assessment of tumor biology. And if the audience could please get their answers in, this last poll question will be closing shortly. And once again, this poll question is asking, what is most informative for your studies? And it does look like majority of the audience has voted, so I'll go ahead, close this poll question, and share the results with everyone. And it looks like majority of the audience with 62% voted for tumor microenvironment. Next was 28% with general assessment of tumor biology, and lastly, 10% of the audience voted for neoantigens. And with that, Dr. Newburn, I'll hand it back to you.
Great, thanks, Vicki. Okay, so moving on to challenge 3. So another challenge in biomarker discovery is the complexity of the data and assuring that proper data analytics is used in your study. And this is often a challenge we hear in the field that our customers voice to us about the ability to integrate data from these broad platforms. So I'd like to next walk you through several examples of some of the analytics provided for our immuno-oncology biomarker discovery with our ACE ImmunoID platform. So we provide information on the mutational landscape of the tumor with accurate tumor mutational burden reporting. We also provide deeper analytics regarding neoantigen load as well as the patient's HLA type. And with our newest updates, we can provide a further assessment of the tumor immunogenomics using our Immunogenomics ID that categorizes and reports on key genes to assess the tumor biology for IO. So first, for tumor mutational burden assessment. And so the supplementation that we provide with our proprietary ACE technology extends beyond the traditional cancer driver genes. And in fact, we supplement over 8,000 genes that are not covered uniformly by the standard technology. And this enhancement augments areas where passenger mutations could occur and then contribute to the mutational load of the tumor. So here are a few examples of genes that have benefited substantially from ACE augmentation to illustrate this. So with our broad approach, we enable an accurate assessment of mutational burden, as opposed to having to bioinformatically extrapolate tumor burden using a cancer panel. We use a whole exome sequencing approach to provide total tumor burden. And this is a snapshot of the tumor mutational burden reporting delivered within our comprehensive DNA QC report summary. And this is reported as total non-synonymous somatic mutations and total non-synonymous somatic mutations per megabase. So further analytics are provided within our neoantigen ID, such as deeper mutational landscape assessment by neoantigen load, and also determining the patient's HLA type. And so as you might know, neoantigens can occur anywhere in any gene, including housekeeping genes. And they can also occur in various genomic positions across the gene, so having accurate uniform coverage across the entire gene is needed. And this is demonstrated here with the example of the SYN1 gene. So we plotted the predicted peptides and the various HLAs that they would bind to, so shown here by the various colors and the legend here at the top. And what you can see is that a large percentage of the peptides predicted to bind in this dotted panel are originating from this region, in this area. And as you can see below, this is supplemented by our ACE design, shown here in green, and this area would be poorly covered by the standard exome approach in blue. So this is illustrating that our enhancements can provide an accurate or a more accurate assessment of neoantigen load. And so this schematic shows the workflow for our neoantigen analytics engine. So with neoantigen ID, we can provide overall neoantigen load, as well as give you a complete report of the predicted neoantigen candidates. And so this all starts for us with inputting high quality exome and transcriptome data into our pipeline, right? Because you want to avoid gaps like we just showed in the previous section. And then this shows you an overview of the modules that we've created. So we consider all variant types in our antigen predictions. We look at SNVs, indels, and gene fusion events. We also employ both MHC binding prediction for both class 1 and class 2 neoantigens. And then in parallel, we are using both the gene and the variant level expression data. And then we also incorporate secondary metrics such as similarity to cells, similarity to known antigens, and immunogenicity in the report out. And with current neoantigen predictions, most of these analyses to date have focused only on somatic point mutations, or SNVs. But insertions and deletions could prove to be a very rich source of neoantigens, specifically frameshift indels that produce a novel open reading frame. So as we've seen recently in the literature here from Dr. Charles Swanson's laboratory, when the authors looked at the HLA binding affinities of the frameshift mutations across TCGA samples, frameshift indels resulted in 3 times the number of high affinity binders than non-synonymous single nucleotide variants. And the authors also assessed the association of indels with clinical response to checkpoint inhibitors and detected a significant association of indels with checkpoint inhibitor response in melanoma patients. So indels could really prove to be this rich source of immunogenic neoantigens and also may be potentially a better predictor of patient response. And so it's important to note that the Personalis pipeline includes not only SNVs, but also indels and gene fusion events in our neoantigen predictions. So here are some snapshots from our neoantigen ID to give you a sense of the representation of the report out that we deliver. So we provide overall summaries. We report out on mutational burden. Here, 201 total non-synonymous somatic mutations. We also show you the total neoantigen burden, and also peptide binders found at an allelic fraction of 5% or greater in both the DNA and RNA. And also here's a high-level summary table showing the total number of peptides that will bind with sufficient strength to elicit a response. And here we're using a less than 500 nanomolar threshold. And those that are predicted to bind to at least 1 or 2 or 3 HLA types. And then of those that are predicted to bind, how many of those would be observed to be expressed based on the transcriptome data? And then we also show the binding affinity range and median affinity. So other summaries provided in our reporting include a table with the top 10 neoantigens based on class 1 binding affinity. And this is reported as shown here with allelic fractions of both DNA and RNA. We also incorporate the binding affinities and also our secondary metrics for consideration. And so in addition to these nice succinct summaries, we do provide the full list of all detected neoantigens. So for this sample, I believe it was 399 predicted neoantigens, and so the full list is also provided to your team for some additional investigation and analysis. And also within our neoantigen ID, we provide NGS-based HLA typing using our ACE exome. So as you may have seen in the literature at the very end of last year from Timothy Chan's group at Memorial Sloan Kettering, HLA type is also being proposed as a potential biomarker for immunotherapy. So in this paper, they hypothesized that zygosity at HLA class 1 alleles would lead to a more diverse set of antigens to be presented for immune surveillance. And then, this could potentially correlate with response to checkpoint modulators. So here they looked at over 1,500 patients receiving either anti-CTLA-4 or anti-PD-1 or PD-L1 therapy, and they found that being heterozygous at least one HLA locus correlated with improved survival to immunotherapy. So within our ACE design, we've optimized and augmented the coverage of the HLA genes in our exome and transcriptome. So for example, again, blue shows the coverage using a standard exome assay design, and green shows the supplementation that we provide with ACE. And shown here is HLA-A and HLA-B. And so this enhancement provides uniformity and enables accurate variant calling and ultimately accurate HLA typing. So we have completed a performance characterization of our ability to perform NGS-based HLA typing from our ACE cancer exome. So here we sought to validate hundreds of HLA alleles across both MHC class 1 and class 2. And to do this, we use standard samples from the International Histocompatibility Working Group that had results from orthogonal clinical grade HLA testing or PCR-based testing. And with one of our collaborators, we also performed a blinded study looking at further samples that had clinical HLA data and then again compared to our ACE approach. And so when we compared our NGS-based HLA typing results to the clinical PCR grade typing result, what we found was we had very high concordance. And that's shown here in the slides with our overall results here at the top from this work. And this was for both class 1 and select class 2. So we had high concordance with our exome approach for HLA typing when we compared to clinical grade PCR-based approach. And then in the bottom panel, you can see specifically how we performed at various HLA loci. So again, high concordance with our approach compared to the PCR-based approach. And this represents a snapshot of our reporting. So we provide NGS-guided HLA typing using the normal patient sample that's given. So for class 1 as well as then select class 2. And as you can see, this is provided to 3 fields. And this illustrates how we are really trying to maximize the data you receive from our platform. Eliminates the need to have to send another specimen out for testing, so having to send another sample for PCR-based testing to get the HLA typing results. With ACE Immuno-ID, this is provided in our kind of one-stop shop platform. And so for predicting patient response to immunotherapy, it's being suggested that it's really unlikely that a single metric will completely determine responders from non-responders. And it's thought that, and suggested that a more complex biomarker might be needed in this assessment. So we also enable more multidimensional biomarkers using our immunogenomics ID analytics. So ACE Immuno-ID was designed as a platform that enables multidimensional biomarker analysis, a comprehensive genomic footprint, and an accurate assay. So in the previous section, we just discussed the ability to investigate particular neoantigens or to assess the mutational landscape with neoantigen load or mutational burden. However, we can also investigate beyond this. So you can investigate antigen presentation machinery with this platform. So, for example, looking to see if the tumor is B2M deficient, which can result in impaired peptide presentation. Looking at DNA repair, replication pathways. Looking at immune modulators. So you can look at PD-L1 expression or more emerging targets like OX40, TIM3, or LAG3. Looking at adaptive, innate immune response, chemokines, tumor-associated antigens, as well as cytotoxicity. So as these are all critical areas for IO biomarker analysis. And so with Immunogenomics ID, we provide a very powerful integration of both the DNA and RNA for comprehensive tumor immunogenomics. So we look at various categories shown here to allow for a focused, streamlined assessment of the tumor biology. So here's an example of a snapshot of Immunogenomics ID, pulling together a summary of the key findings in this particular tumor sample. So in categories such as checkpoint modulators, tumor-associated antigens, DNA repair, replication pathways. And so both gene and variant level expression, as well as information on the variant effect and impact are given. So this report out provides a summarized view of the tumor immunogenomics for a quick, streamlined assessment. And a final challenge to consider, and that's often faced in biomarker discovery, is to have the appropriate processes in place with a partner to enable reliable turnaround times for your data. So in addition to innovating with our ACE technology, we've built an enterprise-scale genomics information management system called Symphony. And Symphony orchestrates the many steps in the process and tracks samples from receipt through extraction, sequencing, analytics to final data delivery. And this system allows us to lock down pipeline versions and content for the duration of a clinical trial or a given project. It also enables service in an expedited fashion, following RUO or GCLP guidelines, depending on your particular project needs. And it also allows us to be flexible in the data format reporting and in meeting different data transfer specifications that might be unique to your particular database. So when we engage in a study, our customers are assigned a dedicated project manager that can be that singular point of contact throughout the project. And Symphony allows our project managers to easily provide real-time updates on your sample status throughout the study. So there are other critical components that need to be considered in meeting the operational infrastructure needs of biomarker discovery, and we've listed some of those considerations here. So having the necessary instrumentation and automated processes to meet these fast turnaround times is a top priority. So at Personalis, we have significantly scaled our sequencing operations with the acquisition of the latest Illumina sequencers, the NovaSeq system. So here's a picture from our lab of several of them in action. Other investments needed include the computing power needed for the various analytics that we just went through. So as well as workflows, quality management systems, and the regulatory support through the validation of assays and informatics. So these requirements place Personalis in a unique position as a partner to support the very exciting transformational therapies that are being explored out there in translational research. So in the last concluding slides of my presentation, I wanted to share with you a recent interesting case study from our R&D team at Personalis. And this data demonstrates how our platform can really enable you to identify potential mechanisms of tumor escape. And this data is part of a collaborative work that we've done in partnership with Dr. Sekwon Jang's group at the Inova Shar Cancer Institute in Virginia. And so to better understand mechanisms of anti-PD-1 response, we sequenced and profiled tumors from 19 stage 3 and stage 4 melanoma patients where response was evaluated using RECIST criteria. So of those 19 patients, there were 8 complete responders, 2 partial responders, and 9 progressive disease patients. And the molecular data for each of the melanoma patient samples was analyzed together with the corresponding clinical response to anti-PD-1 treatment. So we assessed the neoantigen burden of these 19 patients and found that overall, higher neoantigen load was associated with a better response to anti-PD-1 therapy. And that's shown here on the graph from patients with progressive disease, partial response, and complete response. And this is plotted as neoantigen burden per megabase pair. So, however, there were 2 notable exceptions that we wanted to explore further. So you can see here that both of these outlier patients have a high neoantigen burden, but did not have a complete response. So we first explored this patient that had progressive disease, but again, had a very high neoantigen burden, and we found something interesting. So after looking at the somatic analysis further, we uncovered that this individual has 2 likely very damaging class 1 HLA mutations. So we discovered a somatic mutation in HLA-A0201 that was a stop gain mutation, and then also in HLA-B1501, a splice region variant. And HLA class 1 genes, of course, are major members of the antigen presentation machinery, and these are necessary for presenting neoepitopes in tumor cells to the immune system. So these somatic HLA class 1 mutations could likely play a large role in this particular patient's lack of response to anti-PD-1 therapy. So we next looked at the patient that was a partial responder, but again, had a high neoantigen burden. And after investigating the transcriptome results further, we found that this particular patient had very high IDO1 expression. And so it's been shown in the literature that high IDO1 expression can lead to multiple immunosuppressive actions, such as increased proliferation of immunosuppressive Tregs. So for this particular patient, an anti-IDO1 therapy in combo with an anti-PD-1 therapy could potentially improve the outcome in treatment. So in this study, we found highly suggestive resistance mechanisms. And this type of analysis highlights the potential importance of having broad immunogenomic profiling of patients that are candidates for immunotherapy so that these types of resistance mechanisms can be further explored. So hopefully this talk provided you with an overview of the challenges we've seen in biomarker discovery and our approach to address some of these obstacles. And I'd like to, thank you very much for your attention today, and thanks for the opportunity to present some details on our biomarker solutions.
Well, thank you very much for that insightful presentation. Now I would like to invite our audience to continue to send in their questions or comments right now using the questions window for the Q&A portion of this webinar. And we've already received some questions, so I'll go ahead and get started with that. The first question we have here is, you mentioned the neoantigen ID and the immunogenomics ID, but what other bioinformatics deliverables are provided?
Yes, thanks, Vicki. Yeah, so we, I focused on those particular reports, but we do provide full bioinformatics output from our DNA and RNA pipeline. So, you get the raw reads in FASTQ format, you'll get the alignments, the variant calls, as well as we give a very nice comprehensive quality control report, full annotation of the variants, as well as we give annotation files that have been filtered by cancer relevance as well. So, and then also with the addition of RNA analysis, we do provide gene expression output as well as gene fusion detection. So it is a full bioinformatics output.
Great, thank you for that response. And the next question we have here is, what specific amount of sample starting materials is needed?
So in general, we can perform the assays with as little as 50 nanograms. But with that being said, it's quality as well as quantity that's important. So for FFPE slides, we generally will ask you for about 3 to 5 slides that are 5 to 10 micrometers with a 25 millimeter squared surface area. So our goal is to get to, I believe, a 1 millimeter cubed volume of tissue, if possible. We also can, if you have already extracted specimen, we can also accept those as well. So if you send us DNA or RNA, when we receive those, we also perform a QC process and would notify you if there are any concerns on the quality before we proceed.
Perfect. And the next question we have here is, from your case study, what additional insights have been covered? So
yeah, so this is new, recent data, and it's very exciting to our R&D team. And with our collaborators at Inova, we've been continuing to analyze the results further. So one thing is we're expanding beyond those initial 19 patients, and we're continuing to increase the cohort size. To again observe how well the neoantigen burden holds to anti-PD-1 response, but also to look further at these molecular mechanisms like we saw with the 2 patients that were outliers. So again, trying to take a further look at responders and non-responders. So, we look forward to some further results from those studies as well.
Great, thank you. And the next question we have here is, do you always need a matched normal in your process?
So we do need a match normal for certain analyses. So, when you have the match normal, it increases the specificity of your somatic analysis, so it's always more beneficial to have the match normal. So, for example, for our neoantigen ID, to determine candidate neoantigens, we do require a match normal for these analytics. However, we also have a pipeline for tumor-only analysis, and in this case, we use a series of filters and annotations to the variants called in the tumor to approximate what the somatic status would be. So like population frequency metrics, we have a panel of unmatched normals that we use. We look at mutational impact, quality, and other metrics. So, these are particular filters that how we try to informatically make up for not having a match normal.
Great, and the next question we have here is, do you have a recommended tumor content for ACE ImmunoID?
So we, at Personalis, we typically recommend a 20% tumor content from your specimen. So yes, that's typically our recommendation.
Perfect, thank you for that answer. And the next question we have here is DNA extraction from FFPE done automatically or manually?
So we do have automatic places in place. Yeah, so that's part of our scale up that we've done to meet turnaround times.
Perfect. And the next question is, how does neoantigen loading differ from TMB?
Yeah, so for the TMB, we're just looking again at total non-synonymous somatic mutations per megabase, or total mutations or total mutations per megabase. For the neoantigen load, we're specifically looking for those that are the more immunogenic and tumor-specific mutations. So using the neoantigen ID, the analytics I just showed you, we report out on total neoantigen load, or the more immunogenic candidates.
Perfect. And this audience member writes in, what is the turnaround time?
So that really depends on your study need. We do have, for our clinical trial partners, especially those that we are providing services for neoantigen-based clinical trials for vaccines, we do have an expedited turnaround time, meeting a 14 calendar turnaround time for those particular services. And for research use only or GCLP studies, it again depends on the volume and the scale, but typically we're at approximately a 6-week turnaround time.
Perfect, thank you. And this brings us to our last question for today's webinar. And that question is, do you offer these services under GCLP conditions?
Yes, we do. So we have a variety of lab environments. So we offer research use only, GCLP, and also we do have CLIA environment as well.
Well, thank you very much for those answers. We have reached the end of the Q&A portion of this webinar. And if you have any further questions, please direct them to the contact information showing on your screen. And here we have an email address, which is info@personalis.com, or feel free to follow them on Twitter @Personalis Inc. And thank you everyone for participating in today's conference. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is greatly appreciated as it will help us to improve our future webinars. Now please join us in thanking our speaker, Dr. Erin Newburn, and you can do so by clicking the link in the chat box. We hope you have found this conference informative. Have a great day, everyone.